Welcome to Cancer Research. In creating this subject, I aim to give you some insights and some tips and tricks I wish I had known when I had started out as a researcher. Ultimately, of course, we want to help you become the next generation of world-class cancer researchers, finding new ways to diagnose and treat a wide range of cancers.
In the last few years, we've seen state-of-the-art techniques such as single-cell sequencing and multiplex immunohistochemistry allow scientists to study cancer at a high resolution.
In this particular lesson, we're going to discuss how removal of inhibitory factors from this side of the equation can lead to tipping of the scales further in favour of cellular proliferation and growth.
There are three main genetic manipulation techniques that can be undertaken in order to assess the role and importance of various genes and their function in cancer cells. These include siRNA, or small interfering RNA, shRNA, or short hairpin RNA, and CRISPR.
Some viruses, such as Rous sarcoma virus, RSV, induce tumors. Many proto-oncogenes encode components of growth-promoting signaling pathways. A prominent example is the RAS-MAP kinase pathway, which promotes proliferation by activating expression of the cell cycle gene cyclin D1.
You'll practice applying these in a simulated clinical case scenario about a patient with lung cancer, where you'll be asked to make decisions about how you might use some research methodologies to further investigate the cause of the patient's cancer, and to investigate in the laboratory what treatments might work treating this specific patient.
So putting this all together now in a real-life example rather than a generic example, here we have a ligand binding to a cell surface receptor. This then activates the RAS-MAP kinase pathway, and ERK enters the nucleus, stimulating the recruitment of the appropriate cyclins and progression through the cell cycle and ultimately cell proliferation.
Today, I will discuss one of the most exciting technological breakthroughs in cancer research that has been rapidly translated into the clinical arena, and that is the application of liquid biopsies.
As you work through the three lessons, you'll help to answer these questions for Jane. Good luck.